Hi everyone, thanks a lot for the invitation, and it's great to be part of this symposium today. And my talk is going to be on effects of autonomic activity on brain dynamics during sleep and wake. So the overview of my talk will be on bold fMRI, covariations of physiology and brain-wide fMRI signal, and then I will talk about the sleep and sleep stages and discuss a little bit on the findings on a multimodal imaging study, which is an overnight sleep study. And then we'll discuss effects of autonomic activity on spatio-temporal brain dynamics across full sleep cycle, including CSF dynamics. So to start with, as we all know now, the bold fMRI, it's a response to a brief local neurovascular dilatory event resulting from a combination of cerebral blood volume and a blood oxygenation effect. And signal results from primarily local blood flow, response to changes in neuronal activity, and we know that it's mostly localized to gray matter. And we are here interested in various mechanisms behind that bold fMRI, including physiological variations. So bold fMRI and physiology. Let's say we have a scan at a tree test, and we have a subject, a healthy subject, and we ask the subject to keep focus on the little central dot, like keep your eyes closed but don't fall asleep, and various other tasks we give. And our mental state also changes with our physiology, like autonomic functions such as control of respiration, cardiac regulation, and also blood pressure. And they basically affect each other, such as the mood ourselves, like if we are awake, drowsy, or sleepy, or too relaxed, or too stressed due to the task we are performing. And basically, autonomic functions and our mental state do interact with each other throughout the scan. And that is also true for the sleep. And then before switching to sleep, I will give a bit introduction on that. So various sensory and physiological stimuli can affect cardiovascular and respiratory activity through various pathways that may include or bypass subcortical regions. And in that review, we nicely discussed that. And because physiological contributions may also depend on arousal state, their influence in fMRI studies of sleep could be particularly difficult to account for. And because during sleep, our physiology and hormones and all the moods has been changing throughout the night, so the circadian rhythm. So I will just go through the circadian rhythm and the sleep briefly here. So it's referring to the 24-hour internal biological clock, regulating various physiological and behavioral processes in living organisms, basically. And those physiological processes are influenced by the rhythm, include sleep-wake cycle, body temperature, hormone production, and metabolism. And as we see, from midnight till morning, noon, and the evening, the blood pressure changes, and then it is decreasing first and increasing again towards the waking up. So, and then basically also to keep in mind, disruptions to the circadian rhythm, such as like during shift work, jet lag, daily routines can basically affect our health and well-being as well. And overall, that rhythm plays an important role in regulating various mechanisms and also physiological mechanisms, which in turn affect our fMRI or brain dynamics. So EEG and sleep stages is, I think, is a critical part of that of sleep studies, and I will just give you a brief information over those stages. So basically, stage one. Let's start with this figure. Gives stage one, stage two, and the stage three. So stage one is the first stage of non-REM sleep, and it's part of light sleep. And it's where we start to slow down, basically. And brainwaves slow down from alpha waves to theta waves, and we decrease our muscle tone, and there is decreased reaction to any stimuli. And then the stage two, then the brainwaves slow down even further, and the body temperature and heart rate even decrease, and that is typically occurring after like 20 minutes we fall asleep. And it's a state of deep relaxation, and also becomes more difficult to wake up in comparison to stage one. And there are a couple of two main markers of stage two that I will also discuss later. One is sleep spindle, and the other one is the K complex. So sleep spindle, you can see it's a distinctive pattern of brain activity, and it's typically around 12-14 hertz and lasts for about 1 to 2 seconds. And K complex, on the other hand, it's another pattern occurring during stage two, and unlike spindles, K complexes are larger and slower brain waves, and its frequency is around 0.5 to 2 hertz. And but basically both are incredibly important for like memory consolidation and cognitive processes. So coming to stage 3, it's also known as a deep sleep or slow wave sleep or even delta sleep. So basically it's the stage of still non-REM sleep occurring after stage 2, and during deep sleep our brain waves slow down even further. From the EEG signal you can also see, and the body experiences a state of deep relaxation. And the amplitude of those slow waves are now like frequent, really frequent, and around 0.5 to 2 hertz as well. And during basically the last one is the REM sleep, so rapid eye movement is this. It's really important where we observe increased heart rate, blood pressure, and breathing rate, such as wake basically. And this is where we dream. So despite the increased brain activity, muscles of the body are relaxed, which is why REM sleep is called paradoxical sleep. And REM deprivation is basically also creates like negative mood, impaired cognitive functioning, and difficulty also with memory retention. So it's such a critical part of our sleep cycle as well. And how we understand if it's a REM sleep, for example, here we observe sawtooth waves, and also we observe eye movements, right? Where the name also comes from. So coming back to the physiology, so it's really critical to collect physiological data during both sleep and wake. And a couple of measures we can have during fMRI experiments, such as respiratory bellows and pulse oximeter, and also blood pressure. So we are collecting those physiological signals basically to understand our signal better and during our fMRI scans. So there has been various papers on like respiratory variation, heart rate variation, and fMRI, and I just chose couple like 3 of them. On left is a work from Pinar et al. From 2006, and this is a resting state study where we see percent of time series significantly correlated with respiratory volume per time. And then on the right is the deep breathing task by Bright et al. And this is also a correlation map where we observe a positive correlation in gray matter, while a negative correlation with the white matter, where we have this gray matter ventricular contrast in the brain during deep breathing between basically RVT and the fMRI. And on the right, those are the maps of person signal variance explained during resting state and from 3 different subjects, respiratory volume and also heart rate. So we see that how those variants explained are highly clearly visible across mainly gray matter regions. So there also has been a work by Whittaker et al. And in this case, they also they use blood pressure, which is also an MR compatible device they are using, and they showed mean arterial pressure and fMRI correlation maps. Basically, on the left the Z scores, and on the right also the correlations as a function of lag. So basically, they demonstrated a significant correlation between blood pressure and both signal, and show that they have a systemic physiological origin, highlighting the fact all localized all signal changes necessarily reflect blood flow supporting the neuronal activity. So what other interesting measures we can have? One of them I want to discuss today is another measure from the PPG signal. So basically, the PPG signal is an optically obtained plethysmogram that can be used to detect blood volume changes. And in context of fMRI, it's mainly used for heart rate variation. But we can also detect changes in the envelope of the signal, basically the PPG amplitude, what we call, and that shows us the fluctuations in finger stick vascular tone. And also where there's a drop in the amplitude, it reflects a peripheral vasoconstriction and otherwise a vasodilation. So there hasn't been a large number of studies that used finger skin vascular tone, and here I give 2 examples. And on the left side there are correlation maps of fMRI with pulse height variability, they call, or the PPG amplitude. And as we see from the maps, the patterns of correlation highly variable across subjects, and then that wasn't actually fully understood in that work. And on the right, author used a similar device called functional NIRS, and functional news, this is also a device attached to fingertip, and they show spatial maps derived from independent component analysis, and they show the highest and the lowest temporal correlation with the peripheral data, first being localized around like sensory regions, and the last one gives the negative correlation gives a region from the white matter basically. So switching back to the sleep, so those studies were mostly during wake and resting state and during task. And we wanted to analyze our overnight sleep data and understand the physiological variations and their covariations with fMRI and EEG across full sleep cycle. So this is an example how the transition from wake to deep sleep is clearly visible in an EEG spectrum from stage 1, stage 2, and stage 3 during a long scan or an overnight scan. So here in this work, we aim to understand the effects of sympathetic activity, basically. So what is sympathetic activity that we observe in the physiological signal is the PPG amplitude drop. So during light sleep, we observed those intermittent drops in the PPG amplitude, which were accompanied by heart rate increases and also EEG arousals that I will discuss. And what we have observed is a widespread PPG amplitude and fMRI correlation across whole brain. And basically in literature, an acoustic noise induced arousal, for example, during non-REM sleep was discussed. And what they showed was an EEG arousal was accompanied by a PPG amplitude drop. And then we wondered how if this was visible in our light sleep data or not. And then we compared our EEG data with the PPG signal, and nicely we observed high co-occurrence of those spontaneous subcortical arousals, so the K complexes I briefly mentioned before, with episodic drops in finger signal vascular tone during non-REM 2 sleep. And as a reminder, a K complex, as told you before, is a signature of subcortical arousal, and it's a marker of stage 2 sleep. So basically, we then further cross-correlated low-frequency EEG power, which is the marker of K complex, PPG amplitude, which is the marker of sympathetic activity, and fMRI signal in whole brain gray matter. So we compared those three signals, and here is like cross-correlation plots between those EEG power, PPG amplitude, and fMRI, and on the leftmost plots, the 0 point we can consider as the K-complex events, which are then followed by PPG amplitude drops and later with fMRI signal drops. And of course, if we cross-correlate those two signals, we obtain a positive correlation in between. And on the right, we show special patterns of correlation at voxel level and for various legs. And we can see clearly that a similar pattern between EEG fMRI and PPG amplitude fMRI, pointing to a common sympathetic origin and giving us the gray matter, white matter contrast as similar to other studies. So coming to the autonomic activity and its effects on spatio-temporal brain dynamics and CSF pulsations. So CSF is a clear colorless body fluid found within the tissue surrounding the brain and spinal cord of all vertebrates, and its main functions are protection, nourishment, and waste removal. And in a recent work, in this study by Fulls et al. In 2019, they observed a coherent pattern of oscillating electrophysiological, hemodynamic, and CSF dynamics that appears during non-REM sleep, stage 2 sleep. And they observed neuronal slow waves that are followed by hemodynamic oscillations, which in turn are coupled to CSF flow. So here you can see in the purple signal, for example, extracted from the ventricle, so the CSF fMRI, they are increasing, so they are increased signals, while the gray matter signals, in gray matter both signals, they observe a drop in the signals. So those are highly correlated with elect behavior during stage 2. So basically, they brought up various other works, of course. This is a study from Alzheimer's disease, and they they showed reduced coupling between CSF flow and global brain activity linked to the Alzheimer's disease-related pathology, for example. And then, so basically they proposed that pulsatile movement of CSF may assist in lymphatic clearance of brain metabolic waste products implicated in neurodegeneration. So we then went further and analyzed our signals across full sleep cycle, and this included like 60 hours of data from 12 healthy subjects. And from stage 2 sleep, so we observed slow waves accompanied basically the K complexes during stage 2 and accompanied with increased respiration and PPG amplitude drops, and on those were ongoing with CSF inflow, as you see in the red plot, and plus global signal reduction. So that was the case during stage 2 sleep. Then we went further and we tried to understand the correlations between fMRI and slow wave activity across full sleep cycle. So starting from deep sleep, stage 2, stage 1, wake and REM. Basically, we observed spatially resolved correlations between slow wave activity and fMRI across sleep stages. And at a correlation of like 6 seconds, for example, where neuronal contributions to fMRI are expected, we observed a relatively strong negative correlation during stage 3, so the deep sleep, and that was prominent in posterior brain areas. While we observed strong and more widespread correlations at a lag of 12 seconds, so this is not really the neuronally expected lag basically, and we thought that this could be mediated by an autonomic mechanism, and this mechanism appears to be most relevant during stage 2 and stage 1 sleep, which are the light sleep. Then we further went and analyzed our data with physiological data regression, and what we have observed basically the correlation of SWA, so slow wave activity, and CSF, and a correlation of, again, slow wave activity and global signal reduced after physiological regression for various stages. For example, correction of fMRI for autonomic signals, so basically respiratory flow rate and PPGM, led to significant reductions in correlations during stage 1, stage 2, and stage 3. But to keep in mind, SWA and global signal correlation lags in excess of 12 seconds found for stage 2 and stage 1 sleep are consistent with a dominant autonomic contribution, while there is a shorter lag for stage 3, deep sleep, which is 9 seconds, may signify a joint contribution from neuronal and autonomic pathways. So clearly, effects of autonomic physiology are important and indeed have not adequately dealt with in fMRI studies. And proper accounting for such effects will likely requiring monitoring full spectrum of available physiological parameters, which may include respiratory depth and rate, finger stick and vascular tone, heart rate, blood pressure, and maybe others as well. So just to give an overview on other mechanisms, variations in systemic physiology during wakefulness may also occur. It's not particular to the sleeps, right? And here are some examples such as painful stimulation or a mental task or a finger vasoconstriction that was studied in the early years, basically, where they observed a finger skin vascular tone, like a drop in the volume of the right finger, followed by a deep inspiration. So we also made a similar study, a deep breathing study, basically during a visual task to keep subjects alert, and we observed consistent increased respiratory variation, respiratory volume, and those were followed by PPG amplitude drops. And in this work, we further used an eye camera to track pupil behavior and observe pupil dilations, which is yet another marker of sympathetic activity. And also with the lag behavior around 14 seconds, we observed fMRI signal drops in sensory regions, which is expected. So when we checked this across all subjects we had for this study, 11 subjects, we observed voluntary deep inspirations are associated with CSF signal increase and global signal decrease, and still the shortening of the delay between slow wave activity and fMRI going from stage 2 to stage 3 suggests a non-negligible contribution of electrocortical activity on CSF pulsations. But this study still shows that that CSF pulsatility may be observed during wakefulness, in addition to light sleep as well. So as the last, we also perform the mental task to induce involuntary sympathetic activity variation. And we designed a cognitive task, which was basically a simple arithmetic task. And we also observed PPG amplitude drops following those tasks, regardless of deep inspirations. And those were also accompanied with pupil dilations. Please check our poster for that. And with that, I would like to thank you for your attention.